Welcome back, everybody, to this fourth session on solutions to roadblocks in this virtual G2MC conference. My name is Jeff Ginsberg. I'm from Duke University. I'm also one of the co-chairs of the G2MC. I share that with Catalina Lopez Correa, who is also co-chairing this session with me, and who is the Chief Operating Officer for Rutin N, which is a Center for Innovation in Colombia, South America. And you'll be hearing from Catalina shortly. But it's a distinct pleasure for me to introduce our opening keynote for this session, Dr. Soumya Swaminathan, who is the Chief Scientist at the World Health Organization in Geneva. I've seen her on a number of virtual venues this week. I know she's extraordinarily busy, so we're very lucky to have her here for a few minutes of her time today. She's a pediatrician from India who has done an enormous amount of research in infectious diseases and its translation to clinical impact, and she's spearheading the WHO's initiatives in genomics, digital health, and obviously more recently, COVID-19. So we're grateful to have Dr. Swaminathan here to kick us off on the WHO vision on genetics and genomics. Dr. Swaminathan.
Thank you. Thank you very much, Jeff and Catalina, and thanks for inviting me to this session. So greetings to everyone. Good morning, good afternoon, good evening to wherever you are in the world. And I'm sorry that if you've seen me too many times in the last few days, I apologize for that. And apologize if I'm repeating myself as well. So I don't know, there's a lot that I could talk about, but let me just say that, from the vantage point of being here at WHO, which I think is an incredible opportunity for anyone to be in this position at this time in this place. I mean, of course, none of us want to be in this pandemic. It's just awful. And the kind of suffering that people are experiencing all over the world and the kind of impacts we're going to see for years and years, I think keeps one up at night. But then to be able to be in this position and to be able to contribute to finding a solution for this pandemic, I think is an opportunity that not many people get. So I'm really very grateful and humbled by that. And also the opportunity to work with wonderful people from all over the world, scientists, academics, researchers, social scientists, civil society, who all want to work together and collaboratively in the spirit of solidarity to try to solve it. So as you know, we launched recently something called the ACT Accelerator. So that's access to COVID tools and technologies, if you like, accelerator. And there's been a global coming together, and you've seen heads of state, philanthropic foundations, private sector, civil society coming together and say, we have to accelerate the tools because that's the only way, by using science and by pooling our knowledge and resources, that we can actually bring an end to this pandemic. So the goal of the accelerator is to bring an end to the pandemic through science and through the development of new tools. Now, part of that exercise is to try to develop platforms for sharing of knowledge, resources, in the spirit of open science, open data, and open collaboration. And of course, there are many different elements to this. There's what we've seen already happening, which is open science, open publishing, and all the top publishing companies in the world and the journals making COVID resources completely open access. And that's been fantastic. In the area of genomics, we've seen platforms like GISAID prove to be extremely useful. Of course, GISAID is a platform that was set up 10 years ago to share influenza sequence data, and I think has over 120 countries participating laboratories, and has stood the test of time and works closely with the WHO Influenza Network as well. And they very quickly created a platform to share COVID genetic sequence data at the request of the Chinese investigators. And on January 11th, we had the first, I think it was 6 or 7 genomes of COVID-19. Today, we have something like close to 16,000 genomes from many different countries in that database, allowing scientists to look at real time at the evolution of the virus, to track where it perhaps when it evolved or human transmission began, and also to look at patterns of how it moved from country to country. We're of course also very interested and keenly looking at mutations that are appearing, and so far, luckily, we have not seen any mutations on any of the significant sites like the polymerase or the protease, which are the therapeutic targets, or the spike protein, the receptor binding domain, which is a target for a lot of the vaccines that are being developed. So what's been incredible to see is the huge amount of genomic data that got generated in such a quick time and shared, and also from quite a large number of countries. But of course, the genomic data is generated from research institutes, and very often they're not linked with public health. And also what's happened is a lot of speculation and interpretations based on smaller data sets, which from time to time talk about these mutations being responsible for different clinical profiles and so on. So there are advantages and disadvantages, but I think the question perhaps for a group like this is to think about how can this huge amounts of genomic data that are being generated, unlike any other past, I think, epidemic or pandemic, how can it really be harnessed to inform both the development of countermeasures, but also perhaps the public health measures that countries are putting in place? And particularly, how can we make more of this happen in the low-income countries, and also for people to realize the value of doing genetic sequence data. We saw in Ebola, for example, that DRC was able to, because they have a very good lab in Kinshasa, do a lot of sequencing. But there's also questions about which particular database do scientists or researchers or academics prefer to use, and what are the rules or terms and conditions of a database that are conducive to open sharing of data, but at the same time, protecting some of the rights that people have. Because one of the things that holds back researchers from sharing data openly is the fact that they might lose the right to then use it for future publications and things like that. And so a few years ago, WHO facilitated a statement that came from many different publishers and editors of the top journals, saying that in an emergency situation, we encourage you to share data openly, and that will not interfere or impede in any way your future publication in journals. In the case of completely open systems like GenBank and so on, where once you deposit it, it's for anybody to put to any use. We saw in the case of MERS that the researchers who actually uploaded these sequence data, that some other universities then ended up patenting it and things like that. It became a little bit unpleasant. And so I think it's good for this group to also have a very inclusive discussion on is GISAID, the terms and conditions that they use, the licensing agreements that is that something that would be acceptable? It seems to be acceptable to a large number of people. Can that be improved, and so on? We are also in the midst of discussions on the Nagoya Protocol, and I think that's relevant as well, because that the Nagoya Protocol was part of the Convention on Biological Diversity, and it talks about the sovereign rights on biodiversity. Now, for perhaps unintentionally, pathogens got included in biodiversity, and therefore pathogen genetic data, as part of now the CBD and the Nagoya Protocol. And that has begun to have some impacts on sharing of pathogens or samples, and potentially it could also on genetic sequence data. So WHO, the member states asked us at the World Health Assembly to actually do a sort of landscaping of what's happening in terms of Nagoya and how has it either facilitated or has it prevented or blocked people from sharing, and has it impacted public health in any way, because we are coming at it from a public health perspective. And in influenza, we have seen that it has impacted the sharing of influenza samples in the last couple of years. We've had several countries from Europe and from other parts of the world which have had delays in sharing of influenza samples because national legislations related to Nagoya are actually interfering with that, or putting in place so much paperwork that it takes months actually to develop the bilateral MTAs. So this is something which is going to affect science, affect public health globally, and I think as scientists, it is our responsibility to find a good way out of this. On the one hand, we want to serve public health, and therefore the speed of sharing, the rapidity of sharing, the completeness of sharing of data, information, samples is important. On the other hand, we have to protect also the rights of the researchers, the population, the country of origin, and because it all started, as you know, with influenza in 2007 when Indonesia stopped sharing the virus because the vaccines that were being developed based on the sharing of viruses were not equitably distributed. So that's a whole area of work now that we're doing for COVID, is to ensure that the vaccines that are developed or drugs that are developed, that there's a fair allocation mechanism, and that these are equitably and fairly available, accessible to people all over the world based on their needs, not based on the ability to pay. So it's a huge area, but something that we will have to grapple with, that we're engaging with member states in discussions on. There's a lot of legal issues over there, but again, I try to keep it very simple by focusing on what the public health needs are. As I mentioned the other day when I was talking to the IHCC and the cohorts, it has to be a two-way street. Sharing has to be a two-way street. It has to be fair. It has to consider everyone's rights as being equal, and that we need to have some kind of globally agreed mechanisms, frameworks that people can look to, which serves everybody. We also look at the ethics of some of the new technologies. So we're looking at the ethics of genome editing. We have a committee that's is discussing this, and will in the next few months, I hope, come up with a framework which looks at the ethical and social and regulatory and legal aspects of human genome editing, and provides a framework for countries to adopt on regulating this. Of course, there have been calls for moratorium on gene editing, on human, at least germline genome editing, but our role really is to do the normative work on this, to assess where we are with these new technologies, and then the framework that we provide could be as well adapted to other new technologies, emerging technologies like artificial intelligence or other things, again, which are used for health applications. So the other side of that coin is, on the one hand, the germline genome editing obviously still needs quite a lot of research still to be done before we are convinced of its safety and its utility. But on the other hand, somatic gene editing has immediate applications in health, in diseases like sickle cell anemia, thalassemia, potentially even HIV, for example. And there our goal is to see that the low resource countries and settings are also able to adopt these technologies and benefit from these advances, because the burden of disease is quite often in these countries. And so we are very excited by programs like the ones launched by the NIH and and the Bill and Melinda Gates Foundation last year on looking at affordable technologies that would bring the latest in gene editing to trying to solve the problems of sickle cell anemia and HIV. These were the two diseases that was the focus of that program. There's so much talk about precision medicine and personalized medicine in high-income countries, but we are very far away from that in the lower income settings, where even simple genetic diseases cannot be diagnosed because of the lack of capacity. So I think the goal of this community and a very good goal, which could actually be measured, I think, could be indicators, would be the hardware is getting much simpler, it's getting much more affordable, and it could be now available in many countries, but it's the capacity to analyze the bioinformatics capacity and the ability to really use that information to serve the needs of the population that is, I think, still far behind, despite having networks like H3 Africa and so on. When you actually go to a hospital or a clinic and see how much a diagnostic capacity there is, it's very limited. And I think this is true both for human genetics, but also for pathogen genetics. We know that in many countries now that there's, of course, the use of molecular diagnostics for many diseases, but it's still available. In fact, I can tell you that when the pandemic started and we did an assessment of the diagnostic capacities, we found in the whole continent of Africa, there were only 2 labs in Senegal and South Africa that were able to perform the RT-PCR test for COVID. And so within the next couple of weeks, this capacity had to be built. And today, I think there are about 40 countries. But even in these 40 countries, there may be 1 lab that's able to do it. And for many of the smaller countries, they have to ship samples. So you can see that how challenging it gets when you have to use molecular diagnostics in places where this kind of lab capacity is not there. So we have to simplify and platform technologies, cartridge-based technologies, make it much easier because you don't need a lot of manipulation in the laboratory. But we have to think of the different settings in which molecular diagnostics need to be used, and how can we simplify this further. So again, coming from the point of view of serving public health, how do we make these technologies more user-friendly, of course, more affordable, and based on the realities of contexts where you do not have BSL 2 and 3 labs, you do not have continuous electricity, you do not have running water. So you need to think of battery-operated or solar-operated systems that can function without much of sophisticated laboratory infrastructure and handling. So there is, of course, a lot of potential during COVID. I think we should use this opportunity to build the systems. We always talk about preparedness, but as the Director General often says, we go in cycles of neglect and panic. So every time there's something like Ebola that happened a few years ago, there was all the international panic. But then when it settled down, it goes back to the pre-existing levels of neglect of pandemic preparedness, neglect of public health systems. And it's not just the low-income countries. In fact, we've seen very clearly this time around that the public health infrastructure of many high-income countries has, over the years, been neglected to the point that it could not really, it couldn't rise to the occasion, and the health systems got overwhelmed. So it's, the last point I'd like to make is that all of these initiatives need to be multidisciplinary, and they need to involve not just the community, very importantly, the communities and civil society and social scientists as well. And that makes it much more likely that the end result is a good one. And I think that's what I think this group is also aiming to do. And it's very nice to see so many people getting together to discuss the challenges and ways of addressing those. So thank you very much for giving me this opportunity. And maybe the thoughts were a bit random, but I thought that these were some of the issues at the top of our mind. And as we're moving ahead in trying to bring the global community together to address this pandemic, there's really a need to speak with one voice as far as possible, at least the scientific community to speak with one voice on what is really important for global health and what are the concerns that need to be addressed to ensure that there's fairness and equity in what we're doing. Thank you. Over to you again, Jeff.
Hey, thank you. That was wonderful. And I guess I would just say that your messages were extremely on point for what this organization is trying to do. And if I could just ask one question before you depart. You mentioned, particularly in non-COVID times, your organization is providing tools and providing data to be shared. And this group is interested in how do we implement and disseminate novel and important innovations to the groups that need them the most. And I'm wondering if you could just comment briefly on, how do you actually incentivize and get these things to actually be used? It's one thing to host them, but another thing to actually get groups, organizations, and countries to actually take them up and adopt them.
Yes, I think this is definitely a very good question and it is a big challenge because I mentioned that there's sometimes a divide in countries between the health system, the health ministry and the public health services and the academics, the researchers, universities who have the capacity to do this kind of work. And so what happens is that you have on the one hand, the academics doing the academic work and publishing papers. It's not having any impact at all on actually what's happening on the ground. Now, it's not true. It's not only for COVID. We've seen this over and over again in many different areas. And it's one of the things that we need to try to bridge. The trust in science, we've seen in many countries. I saw a recent survey that showed that perhaps it was only in the US, but that there's a very high degree of trust in science and scientists just now. And this is a good opportunity to capitalize on something like that. But I don't have a very simple answer for your question, except that we need to advocate for the fact that all these tools that we have can help. And I think there's a lot of belief now today in IT technologies. Every country now wants to use an app. In fact, it's the other way around, I think, that there's an over-reliance, perhaps, an over-dependence that technology will solve all the problems that public health has. So there needs to be a balance. The mobile apps are not going to solve the problems of contact tracing, tracking, isolation, detection of cases, etc. It has to be a combination of the two. But there is a huge deal of willingness to use IT technologies for that. And so I don't think the same thing has happened yet for genomics. It's still a little bit distant, I think, for most people. They see it as not having immediate applications. This is, I think, a role of advocacy that all of us have to take on and communicate better, perhaps.
Yeah, Dr. Swaminathan, thank you so much. It's really an honor for all of us here to have you presenting in the panel today. I think what you just say is critical for all of us, and it's really our capacity to put genomics and science at the center and to be able to work on a global and not just regional. And as Jeff said, that's one of the aims we have with G2MC. So it's really a pleasure to see the views of the World Health Organization going towards that and giving also the importance you're giving, of course, as always, but in particular for the COVID to emerging economies and developing countries. So thank you so much. And again, thanks for your time.